Good morning, dosto. Today I'm going to, try solve the MCQs that are asked in the previous year of set examination, that is MPPSC or any other set exam. So particularly, this is the 2018 paper that is of the state Gujarat. Okay, 137. So starting with first question, that which one of the following statement is incorrect with respect to nature and properties of lipid. Okay, so here the question is the options are: most natural unsaturated lipids are cis isomer. This can be true. Okay, N-3 and N-6 denotation of unsaturated fatty acid are based on the last double bonded carbon atom from methyl terminal. Okay, N-3 and N-6. So there are two terminal, that is N terminal and C terminal. So N terminal stand for amino terminal, while C for carboxy. So here you can see that methyl like NH2, or sorry, CH2 terminal. So I think that N-3 N something for amino group, so it can't be for carboxyl terminal. So I think the third option is correct. So let me, yes, first answer is C. Now moving on to the second one, how many fragments of linear DNA of 1 kb sizes are obtained upon treating with restriction enzyme having a recognition sequence? This is the entire sequence TCGA. So it has 4 nucleotide and it is of linear DNA. So it will make a cut at each 1, 2, 3, 4, 5, 6. I think the answer is 4. Let me check. Yes, the correct answer is 4. I marked this question on the basis of the number of nucleotide given in the sequence, that is TCGA, that is 1, 2, 3, 4. So the fragments come upon and after the action of restriction enzyme will be 4. Now which one of the following is a Fetch-Mid vector? This is a theoretical question, so the answer will be P blue script KS. How much is the approximate propeller twist in B form of DNA? So this is also a theoretical question. So I will mark the answer as 36 degree because it is completely theoretical, that the twist propeller in B form of DNA like that. Which let me also check this. Fourth, yes, fourth is 36. Now which one of the following enzyme can alter the linking number of covalently closed circular form of DNA? The enzyme that is related with the linking number is gyrase. It is like you have to keep in mind. So fifth is C. Yes, gyrase is the correct answer. Now which one of the following is a mismatched? I think this is a botanical question, so skip it if you do not if you can't learn the these complicated names. I also I also left this out, but in answers I see that fourth option is mismatch. Now polynucleotide kinase can be used for, so 3' end labeling is particularly for RNA. Okay, here 5' end labeling of DNA fragment and phosphorylation of linker prior to ligation, both B and C can be used for polynucleotide kinase. Okay, now the presence of F factor. This F factor is something related with the conjugation, so it indicates the male. This is an experiment of conjugation. Okay, conjugation is a transfer of entire genetic material, so F stands for male. Now which one of the following? So whenever there is a chemical mutations, this alkylating agents is not correct. Base analogues are correct answer. Let me check. Ninth B. Okay, so whenever we talk about, that is 5-methyl uracil, these are base analogues. Okay, cap is a feature of course capping is a poly-A tail, and capping is a characteristic features of eukaryotic mRNA. RAPD marker is a codominant recessive partially dominant. Okay, so it is not recessive, it can't be codominant or partial dominant. These words are somewhat related, so you have to means whenever you are going to read the entire technique, this is a very clear option, that is dominant. restriction endonuclease with the recognize. I haven't read all these terms, but on the basis of reading these options, I mark the correct answer. That is same site of recognition sequences: isoschisomers, isozymes, isocodimers. So I take isoschisomers. Okay. Which one of the following features mismatched? I mark C option because the parent and the daughter strands of newly replicated DNA are distinguished. No, they are complementary to each other. Okay. The correct reading frame is identified. Okay, exon skipping is associated with RNA processing mutation, and the segment of DNA has 30 adenine and 30 cytosine. So A pair up with T, to 30 plus 30 is 60, and similarly with cytosine, that is 60, then 60 plus 60 is 120 is the correct answer. Now moving on to the next questions, that is, a transcription unit is 1000 nucleotide long. If only 15% of this unit codes for protein, calculate the approximate molecular weight of protein encoded. I skip these molecular weight questions because it takes a time. So now, which of the following is separation two polypeptide of identical molar mass? Molar mass, then isoelectric focusing. The K, it's not K EAT or Km, it's KT and Km. So it is a efficiency coefficient constant. Ab enzymes are related to antibodies, so it is catalytic antibodies. So which one of the following is the correct sequence of 2D? Always remember IEF, isoelectric focusing, and SGSPH. Which one of the following is true for mycoplasm? Mycoplasm के लिए सब कुछ true है except they are resistant to penicillin. Free radical means which one of the following does not protect microbes undergoing aerobic condition from free radical? So in case of free radical, this A, B and C, all the three options produces free radicals except hexokinesis, because it is an enzyme that converts your use in your glycolysis, that from glucose to glucose 1,6 phosphate like this. So hexokinase can't produce free radical. Okay, now which one of the following is false with respect to quorum quenching? Quorum quenching is analog of acyl ACM and SAM, lactonolizing enzyme, garlic extract and flavonoid. I think it's I am not sure about this, so I am going to look into my answers. That is D, flavonoid. Which one of the following statement about quiescent center is not true? Quiescent center, this quiescent center is, the central position of root. I heard this in cell cycle, that is G0 phase. Now I am not sure that what they are asking. It occupies the central position of root meristem. It presents apical as well as lateral meristem. It connects to all initial cell of the root meristem. It has most actively dividing cells in the root meristem. It occupies the central. It presents in the apical. So I think it occupies the position where no cell division taking place. So I think it connects to all initial cell of the root meristem, or apical, or no, I am not sure. It's D. Sorry, in quiescent center. Now see the difference, that quiescent center in plant is something that is related to the most actively dividing cell in root meristem. However, in the case of cell biology, in cell of animal eukaryotics, it is related with the quiescent phase. So be very clear, this is the very new thing that I have come to study. Now which one of the following is commonly used for identification of bacteria? This is definitely 16 sRNA sequence, or 18, I think 18 sRNA, I think so. 28, no, it's 16 sRNA. Okay. Now this is a exactly theoretical question and I have studied it, so I'll try my best to give this answer, that indole acetic acid test is bacteria. E. coli is giving it, all 2 and 3 is not giving it positive. So I think E. coli, think option Let it be. Now moving on to the next question.